So today is a long day for me. So in the morning, is our first class of the semester. So I give 2 hours lecture to my instrument analysis class. After lunch, I spent a couple of hours to write the final examinations. So actually, so just at 10 o'clock, I finished my final examinations. I'm glad to have this opportunity to meet all of you. So actually, we have a lot of interaction, collaboration. So when I saw the names, so all the names I familiar, but we have never meet each other. So first, I would like to thank Professor Anwar so provide us this opportunity so we can meet virtually, so meet online. Hi, Dr. Banga. So I think so we have interact so for more than 15 years. Yeah. Uh, I think so. This is a good opportunity. So there is a nice symposium, so we can discuss our mutual research and discuss some future research ideas. So today I'm going to discuss with you about on our research natural antioxidants in plant-derived foods and their effects on human health. So during the past decade or more or longer, so my research group have research fixed on characterization of polyphenol antioxidants and other phytochemicals in American cranberry, Chinese medicine herbs and teas. So this slide shows the beautiful cranberry, but taste bitter. So and Chinese waxberry, so which have very nice taste, much better than cranberry, and the Chinese tea, oolong tea, which is semi-fermented tea. And these looks like so human beings, little kids. Actually, this is a night of Chinese medicine plant. So in Chinese, we call it the Hosou Wu. So that means so it helps to keep our hair black. So the naughty name is Radix polygony multiflora. So this is just some examples. So we have a study. Why are people interested in phenolic compounds? So because clinical studies demonstrate cardiovascular, inflammatory, Alzheimer's, and Parkinson's disease are all caused by the increasing oxidation stress. And epidemiological studies show an inverse association between consumption of plant products and the risk of those disease, so listed in the first bullet. So the total antioxidant capacity of blood, so plasma increases significantly after consumption of fruits and vegetables. So this research also including some of our contributions. Phenolic compounds are one of the most powerful group of natural antioxidants in plants, so including fruits and vegetables. In plants, so phenolic compounds, so actually they are derivatives of phosphate and shimimate in plants. So these phenolic compounds protect plants from some pathogens and harmful UV light and other stress. So they are responsible for the colors and the flavors of fruits. So from the structure aspects, they contain at least one aromatic ring and one or more phenolic hydroxy groups. So in this list, the top part are simple phenolic compounds, and the lower part are the polyphenol compounds. All of them have good antioxidant properties. They have antioxidant properties is because the phenols, so the phenol radicals, have several resonance forms, so which make the radicals relatively stable. They are very good in terminal free radical chain reactions, so and protect so biologically important molecules such as the fat, protein, nucleic acid. So we know many diseases are caused by the free radicals. When we study the antioxidant, so we're also interested in on the uric acid. Uric acid is the end product of purine metabolism in human body is excreted in the urine. So also is the a large most abundant antioxidant in human body. So the low, if the uric acid concentration in the human fluid in the blood is too low or too high, the both can cause the health problems. So the high levels of uric acid usually associate with diabetes, gout, and renal disease. So another compounds, when we study, so the bioactive compounds, so especially when we study their bioabsorption and metabolism. So an important compound we need to study is the creatine. Creatine is a broken down product of creatine phosphate in the muscle because it generates at a fairly constant rate. So mainly depends on the muscle mass. Why this compound is important in the bioavailability studies. So no matter we study pharmaceutical ingredients or phytochemical nutrients, so because we need examine the concentration of these compounds, their absorption and their metabolisms. So that means so we need to follow the concentration of parent compounds and their metabolites. So the absolute concentration of metabolites and the pharmaceutical and the phytochemical so doesn't make much sense, because so as human beings, so we also need to consume a lot of liquid. So drink a lot of water. Some people drinking more, some people drinking less. So if we use the absolute concentration of pharmaceutical compounds or phytochemical compounds, so between the people, so this concentration actually is not comparable. Some people drink more water, even they take same amount of active ingredients, so the concentration would be very different. So we need a reference. We need a normalization reagent. So the creatinine is commonly used as a reference reagent because it is constantly produced. So in the human body, so when we use the pharmaceutical or phytochemical concentration, so as a ratio of this concentration to the creatinine concentration, so then so this ratio is more comparable between persons. This slide shows so the plant. So the plant cranberry and is products, the RPM and is products which involved in our study. Now I'm present. So because for the these bioactive components, so they are if they have the has effect, so first, so they must be absorbed by the human body. So today we are mainly so focus on examination of their bioabsorption and metabolism and the bio effect. So first, we determine phenolic compounds. So develop an analytical method to separate the phenolic compounds in cranberry and in radix poly garlic multiflora products. So then we first examine their absorption after consumption of cranberry and RPM products. So finally, so we test the effects of consumption of cranberry and RPM products on the urinary uric acid. So and see so how so these fruit juice affect the concentration of uric acid in urine and in the human fluids. So first, we analyze the total phenolic content in 20 commonly consumed fruits. So users so the following Jakarta assess, so which is widely used. So I think many of your guys so use the this message to examine the total phenol concentration in plant products. So we can see among 20 commonly consumed fruits, so the cranberry so contain highest, most highest concentration of total phenol. So then another one is the strawberry. So actually, so all commonly consumed fruits so contain certain amount of phenolic compounds. So we further examined the effect of cranberry juice on the breast cancer cells. So we can see, so from this test, so after we incubate the breast cancer cell with the cranberry juice, we can see, so as the doses of 200 microliter, so the removed, so significantly removed the breast cancer cell, of course. So this is just a preliminary test. So after we see, so the cranberry juice contain high contents of phenolic compounds and they have potential to kill breast cancer cell. So then it's important to looking for how these antioxidant, natural antioxidant are absorbed by human being and their metabolism and the effect. So for the instrument, so what we use when we carry out this research, so just common instrument, so the GC-MS, so different GC-MS, and the common HPLC instrument. So to determine the phenolics in the cranberry products. So these slide shows a flow chart of sample preparation. So for different cranberry samples, so for the free phenolic acid, so we just ground or extract from the juice. So adjust the pH. So extract, because we want to use the high separation and identification power of GC mass, because of the GC, so compared to HPLC, GC column is much longer, so the total separation power resolution is much higher than the HPLC. So even per unit length of column HPLC have a high separation, high resolution power, but the OO, so the separation power, so not as high as common GC. So the GC column alone. So to use the GC, so require the volatility. So the phenolic, for the phenolic acid, so they are very polar, so contain polar function groups. Their volatility is not high enough. We have to irradiate these compounds to increase their volatility and thermal stability. So for the total, so we also treat the sample. So the conjugate phenolic compounds from the conjugate forms to measure the total phenolic compounds in the cranberry juice. So to speed up the processes, so besides conventional hydrolysis processes, so we also used ultrasound assistant the hydrolysis processes to speed up the conjugation. So to convert the conjugate phenolics to the free-form phenolic compounds. So this slide shows the GC mass separation, identification, and quantification. So we can see, so these are 20 phenolic compounds. So we examined so their character peaks of the mass charge ratio, using mass charge ratio. So for the identification. So there's the GC retention time and the calibration equations. So all of them are very good. Now we see with the ultrasonic assistant hydrolysis, so we can see we test the hydrolysis kinetics. So with the time, so 15 minutes, 30 minutes. So then we compared with the traditional conversion. Traditional conversion takes over 16 hours. So these, so we can see the gray, the green eye or the convention 16 hours. So we can see actually, so after 60 minutes, so that we get so more or less constant concentration of different phenolic acid. So that means, so if we use ultrasound assistant hydrolysis, so say 6 minutes, so 90 minutes, so can get the results compatible with conventional hydrolysis of over 16 hours. So these will save a lot of time. So why the ultrasonic assistant hydrolysis? So it can speed up hydrolysis processes. So also speed up extraction processes and the efficiency. So because the acoustic cavitation generate after ultrasonic radiation, so the bulb grows and implosively collapse generates high temperature, which help for the hydrolysis and extraction. So this slide shows the quantitative results of 20 phenolic acids we identified and quantified. So we can see, so for different cranberry and RPM products where we can see, so the phenolic acid actually, so the mainly occur as conjugate phenolic compounds. So when we compare the concentration, the free form and the total form, so we can total phenolic, we can see, so only small fraction exists as the free form of phenolic compounds. So this is for the creatine juice and the source of cranberry. To examine the absorption of phenolic after consumption of cranberry juice and products. So we recruit 11 normal healthy subjects. So basically, so they are our students. So after they consumed control amount of cranberry products, so then we collect the blood and the urine sample immediately. So this is the time zero. So then after half hour, 1, 2, 3, 4 hours, so we'll collect the blood. And after 6 hours, so we collect the urine sample again. So for the plasma sample analysis, so is similar to the determination of phenolics in the cranberry juice and the products. So we determine both phenolic acid in the free form and in the conjugated forms. So we also use the ultrasonic assisted hydrolysis and the derivatization to form trimethyl acyl derivatives. So these slide shows so the chromatogram of phenolic mixture with total ion chromatography and with the select ion monitoring model. So for the total ion chromatography, so which we can extract the spectrum. So for the identification for selective ion chromatography, so which can prevent interference, so increase the quantitative sensitivity for the measurement. So these slides shows so the absorption so of phenolics after consumption of cranberry. So mainly in the small intestine and large intestine. So we can see. So later on, our results also shows so the second reabsorption of phenolic acid. So in the plasma, so we can see, so these chromatograms shows so the simple phenolic acid, so aromatic acid like benzoic acid, hydroxybenzoic acid, and ceramic acid, so which we identified in the plasma, so are mainly so these simple phenolic acid. So for the high molecular flavonols such as myrcene, quercetin, so probably because so the hydrophobicity is too high, so it's more difficult to be absorbed by human beings. So that's why so the concentration apparently below our detection limit. This shows the kinetic data for the absorption of individual phenolic acid. So this is benzoic aromatic acid, benzoic acid, we can see. So the concentration, so first increase, actually they have the second maximum. So that means so the some reabsorption from the from the kidney and reabsorbed in the bloodstream. This second maximum so could be also due to the biodegradation of high molecular weight phenolic compounds in the intestine and large intestine. And now we present, so in this slide, so more connected data. So the first, so yeah, is the total phenolics in the plasma. So we can see, so this is from different subjects. So after the drink, after the consume different cranberry products. So the first one is so consumed juice. So the second one froze, and the third one is the consumed cranberry sauces. So we can see so the maximum concentration. So usually usually appears after 30 minutes, between 30 minutes to 2 hours after cranberry products consumption. The concentration range, so for both for 3 cranberry products, so actually, so the more or less, so the concentration range is not very different. Besides examining the phenolic compounds and their concentration in blood and plasma, so this slide shows so the kinetic data. So actually, for the we'll only have basically only 2 points. So it is at the time 0 and 6 hours. So we can see, so in the urine, so after consumption of cranberry products, so for all subjects, so the concentration of phenolic, so phenolic concentration are significantly increased. So that means so the antioxidant capacity also increased. So we will further examine the effects of consumption of phenolic of cranberry juice on the uric acid concentration, which so is most abundant antioxidant in human body. Very similar, so no large molecules identified or measured in the urine. So we find no quercetin and miracetin, even these compounds. So determined at a significant concentration in the cranberry juice and other products. As I just mentioned at the beginning, so because the uric acid is most abundant antioxidant in the human fruits, also these compounds are associated with human health and with several diseases. So it's important to examine the concentration of uric acid in human fruit, and also examine effects of consumption of cranberry juice and other fruit juice products on the uric acid. So to examine so the uric acid in the urine samples. So we take the urine samples, then dilute, adjust the pH. And take the supernatant and use the HPLC and with the UV detector to examine these compounds. So for the HPLC, actually we use the reverse phase HPLC to separate these compounds. So for the reverse phase, so the C18 column, so the people may notice, so the creatinine and the uric acid, they are small polar compounds. So they have poor retention on the reverse phase columns. So we have to use the highly aqueous mobile phase to elute these compounds. Otherwise, so the retention will be too low to be separate. When use the highly aqueous mobile phase, so yeah, it doesn't comparable with reverse phase. So the phase, coast phase, collapse, and the retention loss. So in order to overcome this problem, so we use a gradient elution program. After we elute out or analyze so creatinine, uric acid, and the internal standard. So then we use increased percentage of organic modify, so to recover the retention of reverse phase, sedation phase. So the UV detector, so we have a UV exam wavelength to 5 nanometer. So we can see, so actually we get a very good baseline separation. So you may notice, so later on, we also develop several hydrophilic interaction HPLC method to determine so these metabolites. So so these just shows so the reverse phase and the highly aqueous mobile phase, so they are not compatible. So because the C18 chain, so push the aqueous mobile phase out, so then the retention get lost. So that's why, so after each one, so we have to use a high percentage of organic solvent to recover the retention of reverse phase. This slide shows the cranberry acid concentration before and after cranberry consumption. We can see in most of subjects, we can see the uric acid concentration increased after consumption of cranberry juice. Some people may heard also, yeah. Because the cranberry is very good for cure women's urinary tube infection. However, when the people drink the cranberry juice and consume the cranberry products, some patients complain. After the drinking the cranberry juice caused formation of kidney stone. Well, we know the kidney stone is mainly caused by the calcium oxalate. This is additional information. We'll also analyze oxalate in the cranberry juice. Actually, in the cranberry products, the oxalate concentration is not sufficiently high than the oxalate concentration in other fruit juice. The oxalate may be not a cause of kidney stone after drink a large amount of cranberry juice. However, the uric acid, we know it has a low solubility in water. This guy may cause formation of kidney stone. We can see after drinking the juice, in most of subjects, the uric acid concentration significantly increased because there are no solubility. There could be a source of kidney stone. Of course, to confirm these research, these results, conclusions of more research or clinical research are still needed. For the effect of cranberry consumption on the urinary uric acid, of course, the causes are complicated. Polyphenolic antioxidants absorption could be one of the main cause. The above, we examine we can see the phenolic acid, especially the simple phenolic acid, are easily absorbed by the human body, and the consumption of cranberry juice can significantly increase the concentration of uric acid in urine. Now we first examine the anthraquinose in RPM products. We use the similar techniques. Extract the active ingredients anthraquinose. Then derivatize to form trimethylsilyl derivatives. Use the GC, FID, and GCMS to qualitative and quantitative measure.
Symposium.
We also use the ultrasonic assessment hydrolysis to measure the total
तुम मेरा
concentration
स्टेटस देखना,
of
मैंने
of
यहाँ वो
of,
वीडियो बनाई थी,
of these
बच्चा की पिक्स बना रही थी तो
compounds. So we can say, so this shows the chromatogram of 5 anthraquinones mixture standard. So use the GCMS so for the identification and the quantification. For the now these chromatograms shows so the determination of anthraquinones in the RPM products. So in the RPM extracts, so we identify detect 5 or 5 active components, but in the commercial juice, so which purchase from grocery, so only 2 active anthraquinones so were detected. So this table shows so the concentration of anthraquinones in the RPM extracts. So in the commercial juice, so in the commercial juice, so we can see, so we better so consume so the the radix powder. So the juice actually so doesn't contain much active ingredients. So here's just shown so the total, so the total anthraquinones after hydrogenation. So well, we can see, so after consumption of RPM juice, so the uric acid concentration also increased in the urine. However, so because of the concentration of anthraquinones in the samples we studied, so it's not there is no. So then we can't find so the kinetic trend for the effect on the urinary uric acid. So from this study, so now come to the conclusion. So we can see so the analytic techniques, the GCMS, GCFID, so the good method for the determination of phenolic acid in cranberry, RPM, and other plant extracts. So ultrasonication so significantly accelerates the hydrolysis of conjugate phenolic compounds. So it also showed from our results, so the selective ion monitoring model so has a much higher sensitivity for the quantification. So because it eliminate matrix interferences. So in this study, so we also developed a message for the uric acid and creatinine measurements. Use the reverse phase HPLC. Later on, we develop a message. Use hydrophilic in the reaction liquid chromatography. So where we can see so the phenolic compounds, antioxidant, they can be easily absorbed, especially the simple phenolic antioxidants by the human body and become bioavailable and affect the total antioxidant capacity and the human health. So I finish my talk and thank you for your attention. Any questions and comments? Yeah. So now for the acknowledgement, so I thank all my collaborators, Professor David Ran, Emmanuel Ojedi, and all. So the work mainly carried out by my graduate students and support from different funding agencies.
Thank you so much, Professor Zhu. House is open for the questions. Is there any question from the audience? Thank you so much, Professor Zhu. There is no any particular question.
Okay, thank you.
But I have a question that urea and creatinine model, which you have seen on the HPLC, can we apply it in the animal studies?
For the uric acid, depends. So I think so. A lot of because so the human so lost ability to decompose uric acid. So many animals. So actually, so they have ability to break down uric acid. So that means so uric acid so are not accumulated in many other animal.
Thank you so much.
Thank you, Prof. Thank you very much.
Our next speaker is Dr. Muhammad Iqbal Bhangar. I request Dr. Professor Farooq Anwar to please introduce Professor Dr. M.I. Bhangar.
Thank you. Our next speaker, Professor Dr. Muhammad Iqbal Bhangar. I feel honored to introduce my very respectable teacher, Professor Dr. M.I. Bhangar. I did my PhD under his kind supervision. Professor Bhangar is a leading scientist and scholar in the field of analytical chemistry. He is renowned for his teaching, research, and scientific administration in higher education institutions. He has obtained his PhD degree in chemistry from University of London in 1979. Later, he worked as postdoctoral scholar at the University of Pittsburgh, USA in 19.
I think that's enough. Farooq, we can straight away start. There's some problem in the audio, I believe. Is that? Are you listening my voice?
Yes.
Yes, sir, your voice is clear.
I think
Yes,
that
sir.
should be enough. I think we can start people. You have already shown on the screen my brief CV, so that should be all right, I believe. Should I begin, or I have to? Is there any problem in the voice?
Yeah, there was a connectivity problem.
Okay, if you want,
Yeah.
continue.
Yeah,
Otherwise,
okay. Yeah, I can introduce the remaining part. Professor Banger has produced 41 PhDs and high impact 220 research publications in journals of repute. He is fellow of the Chemical Society of Pakistan and is serving as editor-in-chief of Journal of Chemical Society of Pakistan. Professor Banger has also remained member of various national and international bodies at different times, including International Union of Pure and Applied Chemistry. In view of his contribution in the field of science education, Professor Banger has been conferred with Azaz-e Fazilat and Pride of Performance and Gold Medal by the Pakistan Academy of Sciences, Islamabad. Presently, Professor Banger is working as associated researcher at the ICBS University of Karachi. So I would like to request, sir, please start your presentation. Thank you very much.
yes, okay. Thank you, Dr. Farooq. First of all, ladies and gentlemen, I would like to express my deep thanks to the organizers, especially Professor Dr. Farooq Anwar. Is my voice clear?
Yes, sir.
Okay.
Yes, sir.
Yeah, okay. For inviting me to share my presentation in this seminar, which the University of Sargodha has organized. Especially, I'm really excited to see that even in these days of prevailing pandemics, people in Sargodha University, especially Dr. Farooq and his team, are concerned about the research activities and whatever we can do with the they are doing. So thank you very much. I'll straight away start my work. Actually, this lecture is based on part of my work that we have been doing for the last 3-4 years at the International Center of Chemical and Biological Sciences at the University of Karachi. It's the noble metal nanoparticles, as using them as colorimetric sensors, their synthesis and their applications. As you know, ladies and gentlemen, that nanotechnology is one of the most exciting and growing sciences. Everywhere you go into the other universities or other conferences, and you see the paper, chemists are very much involved. Chemists are very much involved in the nanotechnology in one way or the other. Why? Because it is the most exciting and it's modern science, and it has got applications in chemistry, in physics, in biology, in engineering, biotechnology, health sciences, and whatever you can think of. Actually, in our work, we have done the work to develop simple colorimetric sensors with the help of noble metal nanoparticles. And this work is needed actually, as you know, that the need to develop innovative and formative approaches to synthesizing these new chemicals has increased in recent years. And if you see the papers, especially I'm talking about chemistry, you'll find lots of paper in of these metal nanoparticles and their applications, not only as chemical colorimetric sensors, but otherwise also. And this is because, why? Because this is the This is because of the size, small size of the nanomaterial. Nanomaterial, as you know, conventionally between 1 to 100 nanometers, we call them as nanomaterials. And actually their surface, their shape, their material, that is their nature, and their size determines their enormous characteristics and importance. And why? Because the nano particles have attracted the attention of researchers because their chemical and physical properties are tunable. They can be tuned for a particular purpose. And they have got a unique physical characteristics down to the nanometer level, which are quite different from their bulk material. And these properties may be magnetic properties, may be optical properties, and maybe electronic properties, which induce exciting phenomena in the chemical and biological sciences. So, yeah. And about this little bit about the nanoparticles, type of nanoparticles, you must have noticed that there are a lot of different type of nanoparticles, both organic as well as inorganic. The organic ones like dendrimers, micelles, liposomes, the carbon-based like fullerenes, or the graphenes, the carbon nanotubes, the nanorods, and other components. They have been classified, although they are organic, but they have been classified as carbon-based nanomaterial. And the inorganic one are based on the metal based like aluminum, gold, copper, silver, or metal oxide based like iron oxides. You must be preparing the iron nanoparticle with the help of iron nano-oxide. Simple. One of the most important things into this is that their simplicities. They are simple to be synthesized into the laboratory and used for certain purpose. The colorimetric sensor is a very broad term. You can see it has been elegantly described in a recent publications in material sciences and engineering in 2017 by some scientists. As you can see, the colorimetric sensor over here, there's a joint term which can be used for biosensors as well as chemosensor. Biosensors means those sensor for the biologically active compounds like proteins, like peptides, like DNA detection, for which the colorimetric sensor has been designed. And chemical chemosensors, because they can be used for toxic gas detection, they can be used for humidity sensor, they can be used for heavy metal detections like mercury, lead, and so on and so forth. And also they can be used for a variety of organic compounds, may it be a drug, or may it be a pesticide, or may it be any industrially important compound. They could be used for their detection if they need. So they have elegantly described it as a measurement device, colorimeter, which utilize chemical or biological reaction to detect and quantify a specific analyte or a event. If you see the last 10 years literature, this has been given in a recent publication in Chemosensors, published in 2020. Data from Scopus for last 10 years. Keywords: if you introduce silver nanoparticles as colorimetric sensor, you'll find a number of applications growing with the passage of the time. You see, it started all from 2019, and so although the research work must have started earlier than that one, but the data is recorded is from 2009 to 2017. You can see, and it has, so up to 2019. So there is a lot of interest of the scientists and the researchers into this field of colorimetric sensors. Noble metal particles are used as why particular, because there are other noble metals also, ruthenium, rhodium, palladium, silver, or others. But since gold and silver nanoparticle furnish among all these very superior characteristics with usual useful flexibility, and they could be designed for a particular purpose. And the other thing, as I told about nanoparticle, which is general for the nanoparticle, is that it is excellent tunable properties and unique physical and biochemical properties, and it can be synthesized. It can be synthesized with the chemical reduction synthesis and surface modification techniques. Wet chemical reduction methods, I think some of you must be familiar. You must be doing in your lab. using sodium borohydrate or other reducing agent. And these properties actually benefit the development of novel metal nanoparticles based colorimeters for visual identification and quantification of organic compounds. The difference in properties at nanoscale is because of the two reasons: because of the smaller size, 1 to 100 nanometers, especially preferably 1 to 50 nanometers, because they have got a very large surface area of the same material as compared to that in its bulk form. And below 50, the lots of laws of physical changes, and there are a lot of exciting things that we can see with the nanoparticles of metals, which we cannot see if they are non-nanoparticles. All nanoparticles, you see, have got a typical characteristic excitation coefficient, and they have got a very high molar absorptivity, or we can say molar extension coefficient. Especially, I'm talking about gold and silver, because we are confined to gold and silver because this is the work which we have been doing for a few years, and because of which you can see they have got a strong UV absorption in the UV visible range, and that could be functionalized. The surface modification of nanoparticles actually is extremely important in controlling not only controlling the size of the nanoparticle, but also increasing the colloidal stability of the nanoparticles, and thereby providing it a what you say, a new functionality to react and to detect other metal ions or other heavy metal ions or organic compounds or proteins or so on and so forth. And this surface Prodman resonance actually is a phenomena where these electrons interact with the photons coming from the electromagnetic radiation and oscillate. And this oscillation has got a resonance. Therefore, it is called as the surface Prodman resonance, a phenomena which is very common in these. In this presentation, we'll be primarily focusing. Okay, we'll be primarily focusing on new a few examples from our work based on the novel metal nanoparticles and the detection of certain drugs and pesticides. There are several strategies that have been developed to enhance the functionality of these nanoparticles, particularly by precisely controlling their size, shape, and monodisperse of the nanoparticle. The surface layer can be functionalized with a variety of small molecules, surfactant, and polymers. And the characterization include their characterization. Once they are formed, they have to be characterized, prove that actually it has been formed is through the NMR, through the FTIR, most significantly to the AFM, atomic force microscopy, the SEM, the TEM, transmission electron microscopy, XRD, zeta potential, VET, data analyzer, and so on and so forth. In this context, you can see it is just the basic slide which shows the principle of detection strategies. Is that silver and gold nanoparticle detection is based on aggregation strategy, where nanoparticles produce visual color change when molecules of interest, that is the annihilates, come into contact with them, and when they reaches to a threshold concentration. The ligand, and actually there is a ligand which is actually an anchor group, which is responsible for the attachment to this inorganic particle represented in this. This is a nanoparticle, say for example silver or gold, and it has hooked up with this organic ligand, and this ligand is then attached with another functional group is attached here, organic group, so that it can actually interact with the coming annihilate, which is actually to be formed. And the reason of this attraction is intramolecular interactions electrostatic interactions, the hydrogen bonding instructions, the hydrophobic interactions, van der Waals forces, and so on, which alter the dielectric constant environment of the nanoparticles. So this is actually very common in the synthesis of nanomaterial. If you must have noticed it. These are just this physical and chemical and biological methods. These days, sol gel methods and the plant material methods are very popular because they are green method for preparing nanoparticles, and they are chemical methods, so easily to be prepared. Originally, actually, Tafit method, which was used quite earlier, and gold chloride salt with trisodium acetate as a reducing and a capping agent for nanoparticles, resulting in particle diameter range between 10 to 15. The Prust method involves two-phase method, gold chloride, reduction of sodium chlorohydrate in presence of thiol as a stabilizer to stabilize the ion. Green synthesis involve ionic liquids-based synthesis, which also provide nanoparticles, a versatile platform for novel metal nanoparticle-based colorimetric sensors. If you see the recent trends in the literature in colorimetric sensor, there are different type of sensors which are being made into the which are nowadays very popular, and they are based mostly they are on surface plasmon resonance SPR mediated sensors, the enzyme and mimetic enzyme catalyzed sensors, the fluorescence off and on based sensor, in which as soon as the nanoparticle functionalized nanoparticle sense the coming annihilate, either the fluorescence is off or it is on. It is and ligand receptor binding, photonic structure based, instrumental based. There are different types of ones, but we have done only work on this one. So there are different plenty of opportunities to embark into this research of colorimetric sensors, depending upon your interest and your facilities that you are doing there. There are some examples of the functionalized novel nanoparticles as chemosensors that I would like to share into this. This is an example, typical example of the colorimetric sensor of as a colorimetric sensor using riboflavin by functionalized silver nanoparticle, which are capped with beta cyclodextrin grafted citrate, published in 2016 by Yongbu. Actually, this beta cyclodextrin is a very important, very useful organic compound, which is both it's an oligosaccharide, which is both hydrophobic as well as hydrophilic characteristics. So it is not only industrially important but also extremely important into the research. And it actually reacts with silver nitrate to form beta cyclodextrin citrate conjugates, which helps in stabilizing and functionalizing a nanoparticle. And you can see into this literature, it is given that this is a nanoparticle, silver nanoparticle capped with beta cyclodextrin grafted nanoparticles, and these which had a strong absorption somewhere around 400 nanometers. And this is the one which is shown in the color with the golden color. Now with a pale yellow color, sorry. The when it reacts with the riboflavin, its color riboflavin. This is riboflavin, which is colorless. Its color, it changes visual color from red to from yellow to red, and thereby the not only that the absorbance is certainly considerably reduced, but another beta shift is can be seen at around above 550 or 540 nanometers, so which indicates the formation of this, the detection of riboflavin. This has been also shown into the next slide by the authors. This is the functionalized nanoparticle. beta cyclodextrin grafted citrate nanoparticles. When it react with the riboflavin, the color changes and it forms this complex, which is an aggregated silver nanoparticle, or which we call an aggregated nanoparticle riboflavin. You can see the color of this, and you can see the, so they can be usually they can be seen visually as well, not only through the spectrophotometric, but also visually. There is a change on detecting the presence of riboflavin has been detected, and colorimetric probe showed excellent sensitivity according to the authors for riboflavin for lower limit of detection to 160 nanometers. The other part of our work is 2,4-dinitrophenyl-hydrazine dithyrocarbamate modified silver nanoparticles as sensors for the colorimetry detection of omeprazole. Omeprazole, you must have noticed, it's a drug, antacid drug, comes with the name of Rysic in our country, in Pakistan, and its determination, like the determination of drugs, is extremely important. So there are different HPLC methods, mass spectrometric methods, and other methods for determination. However, these methods, this method is a very quick and a very simple method, which can be done in the laboratory without the help of big instrumentation. Only a simple UV spectrophotometer is required, and the laboratory work is required. So published by our student Saima Jabin, you see this 2,4-dinitrophenyl-hydrazine is a very common commercial grade reagent, which can be obtained in almost every laboratory, which is present in almost every organic chemistry laboratories. When it reacts with carbon disulfide, form dithyrocarbamate. Dithyrocarbamates are very popular complexing agents with the metal ions, and very popular for the determination of metal ions through the, in previous days we have done work for the determination of metal ions through separation of metal ions through the HPLC. When they react with the with the silver nanoparticles, actually they form this as a functionalized probe, dithyrocarbamate silver nitrate probe. Now this can be utilized for the detection of omeprazole. You can see the structure here changes. The sulfur has got an affinity towards the silver nanoparticles, and thereby this complex formed is a very stable complex. And here it is again also shown as silver nitrate with sodium borohydrate react with and forms this which is the DDT-ATC silver nitrate. Now this reacts with the omeprazole, whose structure is shown over here and denoted by this sign, and thereby this reagent is formed, and this absorption is reduced. The absorption at a visible range, bathochromic shift increases. So that detects the presence of the omeprazole drug. Yeah, this is the typical UV characteristics of the silver nitrate nanoparticles, around 400 or so. These are bare silver nanoparticles, but when they are functionalized, that is functionalized with the help of the dithyrocarbamate 2,4-dinitrophenyl-hydrazine dithyrocarbamate, the functionalization takes place, and there is a shift, a reduction as well as a shift of about 20 nanometers of this absorption maximum of this complex, and thereby it has been also used as a sensor for omeprazole, as I said. And you can see the color change here from here to here. There's a visual color change from here to here, and we have tried on different pH, and we found that at pH 8 was chosen as the optimum buffer condition for colorimetric sensing. Generation of a new peak at 550 nanometers here, and the change in color from yellow to pink. All these things actually indicate the formation of the complex, the detection of the omeprazole. In this way, a linear range of omeprazole is obtained from 0.005 to 0.3 millimoles. and LOD is 5 into 10 raised to the power minus 6 moles of this drug. So this is a very simple and excellent method for the determination of this drug. The other work is on the using the polystyrene block 2-vinylpyridine conjugated silver nitrates as colorimetric sensors for quantitative determination of CARTAP in aqueous media and blood plasma. You see, CARTAP is a pesticide, and the determination of pesticide in samples, water or blood plasma, is a lot of work is done into this one, especially through the gas chromatographic method, through the HPLC method, through the GC-MS method, and so on and so forth. So their determination is extremely important. So what we did actually, we tried to use a block polymer, because there are certain polymers which contain oxygen, nitrogen, and sulfur in their repeat units as potential candidate as a stabilizing agent for the metal nanoparticles. Why? Because of their tendency to form chelates with metal ions by donating lone pair of electrons, the nitrogen or the oxygen or the sulfur. Then the secondary interaction, such as pi bonding or the hydrogen bonding, as I said earlier, the van der Waals forces attraction play a vital role in this chemosensing. In this context, you see the CARTAP has been used with the help of polystyrene block vinylpyridine conjugated silver nanoparticles to find out its concentration. The chelating ability of this pyridine ring here of this pyridine ring actually is utilized for stabilization of the nanoparticles. The polymers can be a source of basic nitrogen, nitrogen to chelate the metal ion through the electrostatic interaction. And Sana Rahim, our student, has done this work. And this has been presented here, although it's not clear in one slide, but if you go through the paper, I have given the reference over here. You can find out details on this one. These polymers actually, what are they? They are amphiphilic polymers. The polyvinylpyridine, that is, they are hydrophilic as well as the hydrophobic. The reduction, and they form micelles, and their reduction with the of the silver ions produces a color from colorless to yellow color, and then when CARTAP is added to this one, the color changes. And here why we have determined CARTAP at this place, and we have done an interference studies using different pesticides. And this is clearly separated, the green one, CARTAP, from the other one, so that the block polymer actually show a colorimetric sensor allow rapid and quantitative detection of CARTAP in the concentration range of 0.36 to 0.036 to 0.36, and LOD is 0.06 microgram per liter. The prepared nanoparticle actually has detected the CARTAP. You can see the this has at 428, and this. These are the way in which the pesticide can be determined with the help of a functionalized nanoparticle at a very small concentration, at a fairly lower concentration, and with the help of a ordinary UV visible spectrophotometer. And of course, the characterization of the nanoparticle depends you have to take the help of the advanced instrumentation techniques like SEM or the AFM or the XRDs sometimes, and other one, but their preparation is extremely important, slow. The other example is the use of the polypropylene glycol. This is also a polymer, stabilized gold nanoparticle, and here instead of silver we have used the gold as an F, and stabilized by the polypropylene glycol. The polypropylene glycol is extensively employed as biomaterial for the manufacture of biomedical devices. It is non-toxic, highly stable, fairly hydrophobic, and compatible and biodegradable. The oxygen atom present to this one actually the ether group that is present, you can see in the next slide, act as an electron donor to the to act as a chelate with the metal nanoparticle. In this study, we have done a one-pot synthesis of PPG stabilized silver or gold nanoparticles, which are synthesized by using tetrachloroauric acid tetrahydride as gold source. And this is a stabilizing agent, and sodium borohydrate is used as a reducing agent. This is the ether group of this, which actually provides electrons to the surface of nanoparticles, and this when this shows an absorption at a different place, and when, however, the CEF, which is sapphire trioxane. Sapphire trioxane actually is a drug which is very used for different purposes. And this drug, when it comes in contact with the drug, the molecule aggregates and thereby attaches with the and form polypropylene glycol gold nanoparticle CEF complex here. Cephalosporin, which is shown over here. And for this study, polypropylene glycol, gold, and CEF detection, all this shown over here, we have used in interference studies, because interference studies are extremely important, because you are doing a study in some metrics, there might be some other things which are drugs which are present into this one. For the interference studies, 16 different drugs were tested. Individual addition of equal concentration of drugs did not result in any of the visual color change. However, when cephalosporin, this one is added here, the color changes from red wine to dark blue, and their absorption is shown over here. So this shows that the absorption of this one is not interfered by any of the drug over here. So very clean and simple method which can be utilized for the detection. And the last example is the green synthesis of methyl gallate conjugated silver nanoparticles, the colorimetric probes. As I said, that these nanoparticles can be made through the help of the silver nitrates, through the help of the gold salt, with the help of agents, reducing agents like sodium borohydrate and other compounds also. But these days people are also doing the green synthesis of the nanoparticles. Green synthesis means that they are using plant sources. They are using plant sources which contains a lot of phenolic components which can reduce this, and thereby form nanoparticles. And you must have seen into the literature very many that preparation of silver nanoparticle with the help of the extract from the roots of this plant, extract from the roots of that plant, or stems or leaves of that plant, flowers of that plant. So that is actually so we have also obtained an extract of methyl gallate from a plant source, and used it for the to conjugate the nanoparticles. And this one of our students has published this paper in New Journal of Chemistry recently. The methyl gallate has a structure over here, silver nitrate. It forms a silver nitrate. Sorry, it forms a silver nanoparticle, stabilized by the methyl gallate. Because stabilization and functionalization is extremely important. You must remember in the beginning I have shown you the slide where anchor and ligands are added actually to, and functional groups are added actually. Sometimes the reductant serves as a reductant as the providing the functional groups also, but sometimes additional functional groups have to be added. So what happens actually, this nanoparticle here it is shown again. When gentamicin, a drug, when it comes in contact with that one, it shows a different structure and different color, visual color change of which is because of the proposed structure of the methyl gallate cellular nanoparticle gentamicin complex, which is shown over here. And UV spectrum, if you see the UV spectrum, UV is visual spectrum of all other screen compounds, and like the one which we have seen previously, and methyl gallate silver nanoparticle, the yellow color solution. But on addition of gentamicin drug, the absorption considerably reduces, and another absorption started appearing over here above 500, which is indicative of the formation of the this complex. Yeah, to conclude my talk, I would like to say that there are a lot of things that are a lot of compound work that can be done in the field of chemical biosensors and chemosensors, and which can be used for the biosensing as well as for the chemical sensing. And during, especially last, as I have shown you in my slide, during last decades, nanoparticles, particularly silver nanoparticle and gold nanoparticles, are reported as highly accurate and visible sensors for the detection of several inorganic and organic compounds. The SPR effect and high extension coefficient makes them very useful, ideal for visual props for colorimetric based analytical applications, including visual detection and spectrometry. Nanoparticles, noble metal nanoparticles colorimetric biosensors are user-friendly, as can be seen through the work that I have shown you above. They can be prepared in the laboratory, environmental, to some extent environmental friendly, because they can be used with a green source, as well as through the use of ionic liquids that are also being reported into the literature for the preparation of nanoparticles. So they are inexpensive and do not require complex instrumentation and long work to prepare them. And but however, still some are commercialized, but still novel metal nanoparticles based colorimetric biosensors are being developed under laboratory scales. Transferring them to the on-shelf, that is industrial, or the applied value of the product needs further work. Needs further work to overcome the main difficulties like reproducibility on the large scale, because things are very good at the laboratory scale, but when you go to the large scale, you have to take care of several things, and sometimes things work elegantly at the small scale, but at the large scale, if you don't take care, it might not work. So you have to be very, very careful while taking on the large scale, and because of the consistent shape of the nanoparticle, the consistent size of the nanoparticle, the stability, interferences causing reducing the sensitivity or the selectivity problems of this one. But still, I'm glad that a lot of people are working on this one, not only in Pakistan but abroad also. And our youngsters have got a lot of opportunity to embark into this science, because this does not, as I said, does not require hi-fi equipment or instrumentation. Only the analysis is certainly, and the characterization is certainly important, that you can do by collaborating with an institution like HEA, ICCPS at the University of Karachi, or at Islamabad, or at Faisalabad with big laboratories, and you can do a lot of work. There is a lot of work that can be still done. A lot of work is being done, especially into the food science and technology. Once again, I would like to express my deepest thanks to the organizers and all the participants. So with this, I come to the end of my lecture. Thank you very much.
Thank you so much. It was very valuable and informative lecture, Professor Dr. Iqbal Bhangar. Is there any question? House is open for the questions.
Yes, sir. Sir, I have a question.
Yeah, please.